Hi, I'm Lucille Kuryla, and I'm going to talk about RNA expression in corneal epithelial cells that are exposed to different wavelengths of UV radiation. Our central question is just whether the stress induced by Far-UVC exposure is distinct from conventional UVC. And furthermore, how does Far-UVC even affect biological molecules like we were talking about in the previous presentation? So our approach was to analyze RNA expression to explore the cellular responses because it provides clues about the biological response to the photochemical reactions induced by Far-UVC exposure. So our transcriptomic analysis showed that there are RNA expression differences between the different wavelengths. And we also analyzed the transcriptomic responses and found insight into some specific genes that are related to 222 nanometer exposure and different pathways that are of interest. So today I'm just going to give you a little bit of background on RNA sequencing and our experimental design. Then I'm going to talk about bioinformatics analysis methods and just a quick interpretation of them with our data. After that, we'll use some of those methods in a different way to support our main hypothesis that 222 nanometer and 254 nanometer light induce different responses in the cells. So starting with the background, I just wanted to do a recap, just a little refresh on how cells work. So we have our DNA in our cell, and then RNA is the messenger molecule that goes to the proteins, and our proteins are what make cells work. So looking at the messenger molecules about how the genes are being expressed can tell us information about what's going on in the cell, which is why we're interested in looking at RNA. So our objective was to compare the transcriptional responses to corneal epithelial cells, and we decided to look at 222 nanometer, 254 nanometer, as well as UVB and UVA, just because we wanted to have like a broader set of results. And by analyzing the differences, we wanted to elucidate the mechanisms underlying the Far-UVC induced damage, and again, gain insights into the Far-UVC photochemistry, because we can then use these insights to determine which safety endpoints would be appropriate for safety studies. So the model we chose was corneal epithelial cells in culture because UV exposure to the eye is definitely a human health concern. The cell line that we used is very similar to in vivo corneal epithelial cells, and we used it as a monolayer of confluent cells, which we exposed to UV in a standardized way and let recover for different periods of time. But this allowed us to minimize external variables by having essentially clones of the cells so that we can reduce the biological variability and have more accurate data where we're only looking at, our insight is that the response to the UV as opposed to some sort of biological variability. Just our experimental design was to take the cells, expose them to different wavelengths of UV radiation, allow them to recover for a certain amount of time, extracted the RNA, sent it for sequencing, and then analyzed the data. So what's RNA sequencing? It's a technique to assess about a snapshot of the gene expression at a given time. It reveals the sequences of RNA transcripts that are floating around in a sample when it's extracted, and then those are then aligned to the human genome. It gives us a comprehensive view of the active genes so we can understand cellular response at a molecular level. And we end up with, we end up with a matrix that tells us for each gene, how many counts of each RNA transcript are present in each sample. And this is what we use for our analysis. So let's just go over, there are three sort of categories of bioinformatics analysis I want to go over today. The first one is unsupervised analysis. Unsupervised analysis is really important because the statistical techniques in machine learning are implemented in a way that isn't aware of the outcome of the groups that the samples belong to. So we can really see if there's like a naturally occurring similarity or separation that exists purely from the data. So the first one I'm going to talk about is principal component analysis, or PCA, which is a dimension reduction technique. In this picture, each dot represents a sample, and the colors are their exposure. It allows us to visualize patterns and groupings in the high dimensional data. The low dimensional space, in this case, it's a two-dimensional space, The first principal component shows 63% of the variance in the sample, and the second is 13% of the variance. Basically, this is used to visualize the RNA expression patterns and whether they're similar or different. And we can see that the purple, which are the Far-UV samples, are all grouped together. The UVC is grouped together, and the UVB is grouped together, and they're all separated from each other. And the UVA and control sort of overlap, but they are separate from the other UV wavelengths, which means that this, and this probably have different expression patterns, and these probably overlap. Another unsupervised analysis method is called hierarchical clustering. It results in a dendrogram visualization that can show natural branches in a tree, so we can understand the relatedness of the samples. The natural branching points group together different wavelengths that indicate similarity between the samples. So here we have similar results that say the same thing as the PCA, where we have the UVB grouped together. You can look at 4 branches here. The UVB is grouped together. The control and UVA are the second branch, and they all overlap. The third branch is the UVC, and the fourth branch is the Far-UV. So they naturally separate through the data. So just to recap, this data-driven, consistent result provides evidence of differences in gene expression between the two groups. As I've said, they agree, as the clustering in the PCA and the branching in the hierarchical clustering follow the same pattern, which gives us confidence in our results. The next analysis method I'm going to talk about is the differential transcript expression, which basically compares the average number of transcripts expressed for each gene between the two exposure groups, resulting in the ratio, which we refer to as a fold change. So if these are genes and the squiggles are transcripts, we can look at the two groups. If we look at the first gene and it has the same expression, then we think, okay, that gene probably isn't relevant to our exposure because there's no difference between the unexposed and the exposed. But in the second gene, there's no exposure, there aren't any transcripts expressed, but there are a lot of them in the red group. So it seems that the exposure probably activates that gene, which is sort of a hint that we should look at that gene more closely. This, you can just visualize it here, where like in one group, if each dot is the count of number of genes, these are clearly very separate. There are a lot of transcripts expressed here and not many expressed here, and this would be considered a differentially expressed gene. So we end up with a list of all of the genes and the log2 of their fold change, as well as a p-value that tells us how certain we are that the differential expression is significant. So for example, ATF3, which is a common transcriptional factor gene that's activated as in a stress response, is very differentially expressed with a log fold change of 8 and an order of magnitude p-value of -255, which is adjusted and very large. And just graphically represented here, this is just a list of all of the genes to see if they're differentially expressed and how differentially expressed they are. One way to visualize this is a volcano plot. In the volcano plot, each point represents a transcript. So our ATF3 is there, and each one of these points is a different transcript. You can think of it as like a gene and how it's being expressed. So the horizontal axis represents the log2 fold change and tells us, the magnitude of the differential expression. And if it's to the right, then it's a positive log fold change, which means it's overexpressed in the exposed group. So it's sort of triggered by the exposure. And if it's, that's right, to the left, then it's a negative fold change, log2 fold change, which means that it's underexpressed in the exposed group. Every single one of the red dots is a differentially expressed transcript. So in this case, there are, I think, over 1,000 differentially expressed transcripts. Another visualization technique for the differential transcript expression is a heat map. And each column, I keep pressing the button. Each column here is a sample. And then each row, they're very skinny, is a differentially expressed transcript. And then the color is the relative expression of the transcript that's like z-score normalized. So we can interpret it to say, oh, this is all the same group, and it's the opposite direction of the other group. So each gene between the two groups indicates like a very consistent differential expression. It's just a visualization method. A third type of analysis that we do provides biological insights, and they're important because, before, in addition to exploring the differentially expressed genes one by one, like we did before, we can look at them as a part of a larger whole, which gives us to see how they interact, which gives us more information about the biology. One of these, so the insights include gene ontology and pathway analysis that can tell us, again, more about the biology. When we interpret it, we want to see what the changes in the expression actually mean for us and which pathways are affected. Some pathways that can sometimes be affected are proliferation, cell death, or stress responses. There are a lot of them. So just one of the things is called functional enrichment analysis. It involves either gene ontology, which is biological processes, molecular functions, or cellular components, or pathways, which come from various databases online. And it's basically just a Fisher exact, and it determines whether more of the genes are differentially expressed in a pathway or ontology than would be affected by chance. Then you can do an active subnetwork enrichment analysis, which is a little bit more relevant because it looks at the clusters of genes with collectively altered activity instead of just putting them in buckets. So it's looking at groups with known interactions and coordinated behavior. So it's really critical for understanding complex cellular processes. One more thing that we can do is make these visualizations, these diagrams of enzymes relevant to different pathways. This is apoptosis, and each of the little boxes is an enzyme in the pathway, and they're colored red if they're overexpressed, green if they're underexpressed, and gray if they aren't differentially expressed, and they're actually shaded. So some of them are darker red or darker green, and we can just look at that and get an idea, about the expression of genes that encourage the pathway or if they're inhibiting it, and just provides insight into which parts of the pathway are being activated or suppressed. So some of these methods can be applied instead of to just the exposure versus the control to give us sort of ideas about the effect of the exposure, to actually show definitively that there is a difference between the 254 nanometer exposure and the 222, just by looking at the results of some of these methods. So we can do the unsupervised analysis, and we see in the principal component analysis of very significant separation on the first principal component, which actually is 75% of the variance in the sample. So that's just more support that they're different. The hierarchical clustering also usually results in two branches that are consistent, and the correlation heat map looks like it separates. And then we have the differential expression analysis, where we look at the genes that are expressed in UVC and the ones in Far-UVC, and then compare them to each other. And we see that there exists differentially expressed genes. So there exists a difference. There are a lot of differentially expressed genes that can be visualized in a volcano plot or in a heat map. Another sort of clue and evidence that there is a difference between the two wavelengths is that when the genes are differentially expressed, they only have 423 that overlap, which are probably just like the generic stress response genes, but over 1,000 of them are only expressed in the Far-UV, and over 500 are only expressed in the UVC, which means they just have different responses. Different genes are being expressed. And this is a helpful figure to look at, when you have more time. But if you look, I mean, but I can switch it back and forth, and you can see, right, the colors are changing, so if you want to look at it when you have more time, you can get a little bit of an idea of the different pathways, the different parts of the pathways that are being inhibited or like encouraged by the increased or decreased expression of the genes. So there's another difference, something you can look at. So just to recap, there's separation in the unsupervised analysis. There is a differential expression, and the genes that are expressed are different. So we're pretty sure that the expression is different, which I think we all accept. So in conclusion, there are distinct RNA expression patterns, and we've found some biological pathways of interest that hopefully will give us more insights into the photochemistry when we look at the way that the cells respond to the different insults. This is really significant too for safety assessments, because we want to understand the expression differences, which would say what type of damage occurs, which would mean what type of assay we need to use in the safety assessments. For example, we have, we're doing the corneal epithelial cells right now without any tear film, but the next step would be to use a tear film and say, oh, these endpoints that would, indicate damage don't happen or do happen, whatever it is with the tear film there. So we need the right endpoints for our assays. And in the future, we're going to validate some of the genes with qPCR, keep looking at the mechanistic aspects of the Far-UVC chemistry, and we just got some more sequencing data back at a different time point, so we can look at the early and late expressed genes in Far-UVC to get an idea of like a time course of how the cell is responding. So just thank you to everybody in the Kleiman Lab and the Brenner Lab for all of their help, and I'm done.
Okay, we have time for just one question, and then there would be time for discussion, I hope, after. Holger.
Mm-hmm.
One question: what were the dose levels that you applied for UVC and regular UVC?
6 millijoules per centimeter squared.
6 millijoules.
Mm-hmm. Oh, okay.
Okay, we have time for discussion later, hopefully.
great.
And now.